Hello, everyone, and welcome back for another week of Growing with My Fellow Growers. I'm your host, Jack Greenstock, joined as always by an amazing panel. I'm going to pass it over first this week to Spartan Grown. Welcome back.
Thanks, Jack. Hello, everybody. I'm Spartan Grown. You can find me on Instagram at Spartan Grown, one word, all no spaces. And if you don't do Instagram, you can shoot me. The best way to get a hold of me is through email at spartangrown@gmail.com. And I can help you with all your organic or synthetic cannabis gardening questions.
Happy to have you back. And we got We Nerd DWC first in the chat this week. I just clicked on over to the live chat myself, reminding anybody if they want to see all the messages, go on over there. If not, you can hang out in the, whatever top chat. I think it might be all messages versus some messages now. But next up, we've got Dr. MJ.
Hey guys, I'm Dr. MJ Coco from cocoforcannabis.com. I want to make sure everybody knows about the upcoming spring autoflower challenge that we're doing over at Coco for Cannabis. We're doing size matters, and we're going to try to grow the biggest or the smallest autoflowers. So that should be a lot of fun. Hope you guys can join us for that. And I do the Ask Dr. Coco show every Monday night for my Patreon subscribers. So looking forward to the show tonight. Although I'm behind, I didn't do my homework this week, so I feel guilty about that.
It's all right. I think sometimes looking at it fresh can help us all go through it. And like many of the people in the chat who haven't probably looked at some of these papers, it's nice to be able to go through them and maybe tear them apart or, agree with the science if it's being done well. So I'm looking forward to it because I've only kind of skimmed the surface, admittedly myself, but I know Matthew's got some notes. And speaking of, Matthew Gates.
Yeah, no, I look forward to it. I like when we do these analytical, paper readings that we do. There's a lot of interesting stuff, of course. And yeah, if you don't know, my name is Matthew Gates. I'm an IPM specialist. So if you're interested in more information about that, you can check me out on my YouTube channel. I'm actually going to drop a ultimate russet mite video, probably the best reference for russet mites ever. I'm going to say it online because there's not a lot of people talking about it. So you can check that out over there in a couple of weeks.
Looking forward to that. Definitely a tough one to fight and good to know the best info and most thorough stuff about that. Next up, we've got the American one.
Hello, Jack panel and everyone in chat. I am the American one on YouTube and the American one _with _Keens on the IG. It's always good to be here to see everyone and talk about cannabis. So I'm glad to be here.
Glad to have you here as well. And I'm hoping that my internet is going to be stable, although I'm a little bit concerned that I might be dropping in and out because I'm looking at my TV, which has like a Chromecast plugged into it, and the Chromecast is resetting. So I'm like, is that just the Chromecast or is that my internet? And let's see how long it hangs on for. But with that all said, I know that we have two articles that we were kind of looking at in our little chat discussion. And Matthew, I think, made some notes on maybe one or both of those articles. So I guess I'll let you pick because you put some time and effort into it, which one you would like to start with, because they're kind of somewhat seemingly related, at least as far as I wrote microbes in the title and scientific papers. But as far as more information, I think you'd be the one to kind of lead us off on this.
Well, thanks, Jack. I think that we should do the one Tao suggested last time, or when we were suggesting these, which is cannabis seedlings inherit seed-borne bioactive and antifungal endophytic bacilli. I actually read this earlier in the past, so I'm somewhat familiar with the topic, and I can share my screen so that it's a little bit more engaging for people.
I know I've heard people anecdotally report in the past about stuff like this, so it's always interesting to see what the research has to say about it.
Yeah, as you can see here, my tabs, I'm doing a lot of russet mite reading lately, so a lot of cool stuff to come. But yeah, here it is. Here's the paper. I highlighted a few parts of the paper just so people know what's going on. I think these are just kind of the fun things. We'll go over this diagram here. Basically, this paper in a nutshell found a bunch of endophytes. Endophytes are microbes that are inside of a plant. Then you have a term like epiphyte for microbes that are on the surface of the plant. So that's the difference there. And some of these endophytes are passed from their parents to their offspring. And people who have watched us know this and have heard us talk about it all the time. But Tao suggested a really great paper. I'm not going to read this whole abstract, but this highlighted part says that both hemp and marijuana, so they make a distinction here, seedlings inherited a closely related group of bioactive endophytic bacilli. Actually, I'm going to increase this to 200%.
And Matthew, is bacilli just a plural for like all the bacillus?
Yeah, it's a little bit ambiguous here with the way they've italicized it, but that's my interpretation. So like bacillus and I guess bacillus adjacent, unless somebody else has an understanding different than me. That's kind of what I think they mean. That's how I could tell. At least that's
Yeah,
from what
it's
I've
just a
read
plural
here.
form. It doesn't necessarily mean, I don't think, all of
Like
them.
a genus.
Yeah.
Yeah, but they italicized it. Yeah, I just feel like that's not in keeping
They
with
italicized
the.
it because it's Latin.
Yeah, but it's not a genus. So I guess that's the confusion that I'm having. I understand.
Bacillus isn't a genus?
Bacillus is.
Cannabis is also
Bacilli, I mean,
italicized.
plural
You would
of
just
bacillus.
say bacillus though. Yeah, I don't. You know what I'm saying?
You're referring to several different sort of groups of them.
Well, we don't do it here with Alternaria. We don't do it here with Penicillium. We don't do it here with Cladosporium or Fusarium or Rhizopus. We don't say Rhizope. We don't see Fusarii, right?
Yeah, but in that first thing, it's talking about a group of bioactive endophytic bacilli. So referring to one group of the bacillus genus, actually non-inclusive of the rest of the genus. Yeah, okay, it's a fairly minor. This is a linguistic point more than anything else. Let's get to the rest of it. But yeah,
I don't know if it aligns with the international code of zoological taxonomy or whatever.
sure.
But here we go. All cannabis accessions possess seed inherited Paenibacillus mobilis, and that's a very interesting one. We'll talk about that. With the capacity to solubilize mineral phosphate, they make some inferences that there might be some selection pressure in cannabis that made this a particularly common one across the accessions. Additionally, seeds were found to carry genera of fungal isolates known to be cannabis pathogens and post-harvest molds. Like I said earlier, Alternaria, Penicillium, Cladosporium, Chaetomium. Sorry, I have a little note here that's come up. Yeah, Chaetomium, Aspergillus, Rhizopus, and Fusarium, and many people will recognize several of these genera as pathogens. 13 seed-borne endophytes showed antibiotic activity against Alternaria, Aspergillus, Penicillium, and Fusarium. The study suggests both fungal pathogens and bacterial endophytes that antagonize them are vectored across generations in cannabis as they compete over this shared niche. That niche, the plant body itself. We are all an ecology. So I don't want to just monologue. So should we stop there and have some discussion, or what?
I think it is interesting to kind of note, we have in the past on this show talked about things like hop latent viroid and how 10% or maybe even more were passed through seed, and that's not a beneficial, but it has shown, and you've often discussed in the past that this is possible. So it's just I'll throw my hat in there and say it's interesting to finally see it be put to research and discuss the exact specifics of which ones are being passed and how it can, even though there might be some like pathogens in there, the beneficials are providing some sort of resistance, maybe it seems like.
I think the same is also true for people, right? In a lot of ways, you go first, Spartan.
Well, I had a question just trying to understand this. So everything that they found was an endophyte. So to me, that tells me that's something that would stick around even if you did any kind of a seed wash or anything, because it would be contained within the embryo of the seed. Is that what they're getting at?
Yeah.
Or
yeah,
would
that's
this
right.
be something? Okay, so they're not talking about bacteria or anything that is found on the outside of the seed shell itself that then infects the root.
Well, they
actually
they do. Well, so yeah, that's generally what they're talking about with endophytes, but they actually do. They do one where they don't wash it, and they do find some additional stuff. So that is perspicacious of you, Spartan. Do go over that.
Okay, cool.
That's a good point as well, Spartan, because I have washed seeds in the past, and despite being washing on the exterior and trying my best to like quote unquote sterilize and then hit it with a beneficial, small percentage of them still had a pathogen take within their germination process. So it's it could have just been literally within the inside of the seed, and there wasn't much I could have done. It was going to sprout with that, almost regardless, it seems.
Yeah, definitely. So I've highlighted these two parts here that basically that microorganisms that have effectively colonized plant tissues have the potential to exert that, like you're saying, Jack, this barrier effect against other microorganisms, preventing their colonization.
Yeah, that's often called the founder effect. That, I mean, it exists throughout biology, that the first organisms to colonize a place sort of change the place in such a way that it makes their own colonization more plausible and sort of more difficult to other, bacteria, microorganisms, animals, plants, whatever it is. That's a sort of common thing that we see. The first to get there sort of set up shop and make it their own.
Absolutely. And yeah, like you say, and very much like what I was saying earlier, we are in ecology. This plant is an ecological niche, and so it's absolutely the right terminology to use. Definitely. I think we also get some. If I'm bold enough to say, I think we also get some. What's it called? Niche construction. So that's when an organism modifies its environment. That makes it more like Dr. Coco is saying here, more habitable. So like an ant creating an ant nest, for example, or a beaver making a dam. And then those can have radiating and do have radiating effects on other organisms, right? And it's turtles all the way down at that point.
Yeah, and this happens in like a broader ecology scale. Like the first large plants to reach an island will have a tremendous effect on the flora and fauna that exist on that island forever, because those sort of first large plants, say trees, create the climates and the ecologies for all the other organisms that sort of depend on them, and they become these sort of keystone species that set up sort of the whole ecosystem for them. Other trees will have a much harder time sort of colonizing those islands after that's already taken place. Even after you cut down all of the original trees, that same species will be more easily able to recolonize that area because the whole sort of ecosystem has been tailored to suit its needs. And it's interesting to think about that in all these inheritable things. Like, do you have the same suit of stuff from your parents because of vertical transmission directly, or is it because sort of these other sort of things? Say you inherit some of the microbes through vertical transmission, and then they create that effect on the species that allows other things to colonize that become very similar to what the parents were.
I think it's some nature and some nurture. They are brought up in that region. Then, like in the plant or human individual, they're eating like a lot of the same foods, and they're like pine is the example that came to my head. It doesn't seem to be hospitable to lots of other trees. It changes the pH of the soil, and it does lots of other things to sort of dominate and ensure that it's going to continue to survive. And then other plants and animals that coexist well with pine will then come in, whether it's like a blueberry bush or something that can accept the more harsh pH or other things like that. But that's a great point. And it's interesting that it starts as early as we're seeing now with like the seed, within the seed there on a micro scale, the microbes are fighting to get that sort of founder effect. So very fascinating.
Yeah, no, that's interesting.
My favorite example of this, for those who haven't said, and I'll just be quick, is that there's a saltgrass that gets a fungus endophyte, and that fungus endophyte has a virus in it. And if the fungus doesn't have that virus, which is called the curvularia something or other thermotolerance virus, it might have a number attached to it, that saltgrass cannot exist in this really inhospitably high temperature soil, which is really fascinating to me. So definitely, a turtle on a log on a bump in however that song goes at the bottom of the ocean. It's all connected. So this section here, this says that, like we were talking about earlier, microbial inheritance via seed to seedling, vertical transmission occurs through microbial colonization. There was a section here. Oh yeah, they hypothesize here that cannabis seeds vector opportunistic fungal pathogens as well as bacterial symbionts that protect them against biotic stresses. So we kind of already went over that.
I'm curious, Matthew. So they're saying that there are some endophytes that basically go in and colonize the seed, and then when the seed germinates, they're already there to be, hosted by the new seedling that goes on to become a plant. Correct?
Yeah. So like it's somewhere, it's in their tissue, right?
Right. So I wonder what the shelf life of that is, and if it's exactly the same as the seed, if the endophyte is feeding off of something within the seed during that dormant period, I'd be interested to see some of that, if that makes sense.
I would guess it would drop over time. Like a 5-year-old seed's
Yeah,
gonna
I
have less
agree.
activity than a 1-year-old
Yeah,
or 1 month
exactly.
old.
Exactly.
I think that's a really
follow-up.
I think it's a really interesting follow-up. I agree with that. And I suspect Jack is correct there.
Just as a second.
And is it consuming part of the sort of carbohydrate load for the would-be seedling that's coming up? Is it feeding off of something, or how deeply does that endophyte also go into dormancy?
Yeah, right. Because is it like some sort of like encystment that's like basically torpor and doesn't really do much metabolism? Or is it like what is for all intents and purposes a very small like mycelial mass? And is it intracellular? I don't know. I think most are probably not intra, but endophytes can be crazy.
Well,
Yeah.
I also think about there for a long time, and this I think has pretty much been disproven, but people will still stand by it. People used to put seeds in their mouth, and I think they thought it was because they believed the plant would sense what they were like lacking in nutrition and then grow more of that or something like that. But what I'm thinking now, reading into stuff like this, is it was probably more so when they put the seed into their mouth, they had a certain microbial profile within their saliva that was maybe activating or causing something on the outside of the seed that when it does go to germinate becomes part of that consortium of microbes, and it could be beneficial, and it might actually be more problematic than could be expected, because they're completely different sort of profiles of microbes from what I understand. What we use to break down food in our mouth versus what is most beneficial to germinate a seed, I think maybe there's some crossover there, but I think that Venn diagram is like a narrow scope in the middle. But I would need to look more into it, because I know that's something that people did for a long time, and I want to know more about that side of things now, because I heard it advocated for a long time, but never saw any real evidence to substantiate why or how it would benefit.
I think that you're right that I think there is maybe a very small overlap, but I think it's not zero. I think you're right, though. It's a very narrow Venn diagram. I'm not sure how useful it is personally, but I do think that I feel like there's something there, though. I feel like it's on the track for something interesting, but I'm not sure. I don't have a fully formed thought about that.
Somebody said, what about people who roll their seeds in myco, or maybe like any other? I've seen soaking them in beneficial microbes and things like that.
Absolutely. In fact, I think that should radically change. I'm very excited about the possibility for really fine-tuned microbial consortia that can then be just added to a seed as like a coating, and then have just a really sophisticated barrier, and also sort of trying to use a good word, but like a booster, essentially, something that really helps on multi-tier front. Yeah.
Like an anti-pathogen, but also either releasing more nutrients to the plant or whatever's available, or just making it more protected and active.
Perhaps even for specific effects too, like maybe it's a booster for growth, like just vegetative growth, or maybe something this might just sound like marketing. I'd really want it to be super verified, but
I think a small bit of GA3, like gibberellic acid, if you dialed it into the right amount, or something similar,
or something that makes a plant hormone, that so it could do it itself with just the nutrients you already have.
Or one of the box that we were talking about.
On that in that paper, they talk about making the seed coating with bacillus in particular somewhere.
Oh, I think you might be right there. Yeah. I don't remember
Yeah.
exactly where. Maybe I've highlighted it.
Well, and this is a practice that's kind of standard in agriculture, right? They use like sulfurs or osmocote and different types of, or I don't know, osmocote. There's some sort of fertilizer that is put
Yeah,
on to
seed coating
seeds.
is already a thing. Yeah. So
Yeah. Seeds coated in fertilizer is a very common thing in sort of agronomy writ large. A lot of that, like grass seed in particular, is sold that way.
That's a good point, actually. So yeah, so here, just as a started with the results, I like it when they do that. So they said they had total of 136 bacterial isolates that were obtained, and they used 16S sequencing, which I'm only familiar enough to know that when I look at bacterial phylogeny papers, they often use this. So this is part of the, I think this is different in different bacteria. So although the seeds came from 4 different locations and growing conditions in Canada, they observed an unexpected conservation of some of the culturable seed-borne seedling endophytes, and they have table 1 to show that here. This is table 1. Yeah, this is a nice, oh, that's figure 1. I'm sorry. Might have passed it, whatever. But anyways, getting to the important parts. 15 cannabis accessions appear to be dominated by endospore-forming bacteria. So maybe that explains what we were saying here. Oh, but they didn't italicize bacilli here.
Okay, yeah, that's true. They didn't italicize that.
But here they're talking about a taxonomic class, but so perhaps it was that they were doing the Latinate thing. Notably, Painobacillus mobilis isolates were cultured from endospores of 100% of accessions, despite the different conditions in which their mother plants were grown. And what else? Oh yeah, Bacillus subtilis was found in a majority, 8 out of 15 of the accessions, and was present in both hemp and marijuana. So cool. It's actually, this isn't what they were talking about, but honestly, it's a really cool diagram. So I think it would make for, here we go. Perfect. You guys can see it good?
I can, yes.
So this is where all the different seeds come from. And I like how they like pictured all the different ones, and it shows us like the size and the color. This one looks a little suspect here though, I have to say.
I was gonna say some of those don't look the greatest, man.
I'll say that
No.
if you have 1,000 seeds that you made
Which
yourself,
is why I'm glad they said it.
you could have a representation of every single one of those from like the same, like my Velvet Punch. I've got a bunch, and I could pull out one. I eat the white ones, but
those white ones,
I don't know that showing us
there are a
pictures
lot
of the
them.
seeds. These are the kinds of things. They're fun visuals, but I don't think there's a lot of scientific value in showing us a picture of the seed.
Well, it is if we think that they're using bad seeds.
Yeah, I mean, I suppose that's true.
No, but you're right. It definitely makes for a nice visual to look at. But
Yeah,
I
breaks up
agree
all
with
the
you.
words.
you can
I'm
wipe
not
off
sure
tiger
which one-
stripes, and white seeds will also grow, like Tal had mentioned. I've seen people. I've germinated myself a 15 pack, and 14 out of 15 germed, and they all grew into perfectly healthy plants. So a white seed is often, it might not look as appealing, and some stuff is genetically more higher number of white seeds. So I know some breeders that have put out stuff, and they're like, hey, 5 out of 10 of these might be white seeds, but try and pop them, they'll grow. It's just how that seed ended up, or maybe they're processing them and maybe removing some of that outer coat.
Nope. Okay. I was just wondering if like maybe the authors were like growers, and maybe there was some marketing going on in this research report. Anyways, because maybe somebody sees the tiger stripes and goes, "Ooh, I like how that seed looks. Maybe I'll buy some God's pink." Or
It wouldn't be the
something
first
like
time.
that.
We had like an outdoor
Wouldn't be,
grower
would it?
sponsoring, outdoors better than indoor paper type thing on one of our past shows. So.
Yeah, that's
I
right.
really think this is just somebody trying to dress up their report. I mean, I've been involved in like judging academic posters at academic conferences, and it's the kind of thing that you do. You kind of make something pretty to look at that's related to what you're doing or illustrating some real point, but it's not really sort of moving the needle forward in terms of the science much.
I feel like
That's
they
what
could
I
have
kind of
made
look-
use. They put a lot of negative space here. If I'm going to be judging them on that, they could have made that more compact and made my viewing easier. Anywho, do you know what notobiotic means, Dr. Coco? Because I have forgotten, and I didn't look it up before. I knew I was going to forget something.
No, germinated in notobiotic.
Quick chat. tell us what
Yeah,
that means.
we can figure. I mean, I can find out too. I have a Google.
Let's
machine
crowdsource
here.
it. I'll look it up right now.
We all got the Google machine.
Relating to or denoting to an environment
Notobiotes.
for rearing or culturing organisms in which all microorganisms are either known or excluded.
Okay. So in other words, yeah. So like what they're doing here, that makes sense. Okay. Yeah, so they have a diagram here talking about seed endophyte, epiphytes, and endophytes. So like Spartan referred to earlier, we have seed surface and seed in the endosphere being vectored. And then, yeah, seed inherited cannabis endophytes from seed endosphere and seed surface. So that diagram is showing microbes getting in and around the root and perhaps even the beginning of the stalk here. So that's cool. And then this was table 1, right? So here's the hemp cultivars and the marijuana cultivars, and some of them are indoor. And then we have flax. Okay, so we had the flax seed here. This was a control.
I just want to say a quick note, because earlier I said, why is cannabis italicized? And when Doc was mentioning that it was all of the Latin.
You mean bacilli?
Well, but we were talking about all the italicized words that were in Latin.
Oh, yeah,
and
yeah.
Cannabis is the Latin term for marijuana, because they're referring to it as marijuana. And if you type like Latin for marijuana, it pulls up cannabis sativa. So that's probably why.
Yeah. Yeah,
Well,
and
but it's like
the genus name. Cannabis
taxonomic
is the
Latin.
genus.
Yeah.
So when you're referring to it in a scientific sense, instead of sort of as a casual term, which you can kind of correctly do both with some of those terms. I think cannabis is an example of it. But yeah, that is the genus term too for cannabis sativa.
And sometimes like the name that people use normally is like the genus name, right? Like
Yeah.
I'm forgetting one at the moment, but is zinnia one of them? I don't think so. I'm forgetting. Anywho, yeah.
Can you explain more what we're
things that
looking
are
at
in the
here?
same genus look nothing like each other or behave nothing like each other too.
That's true too.
So yeah.
But yeah, so
Okay.
here we have, oh, what's up?
That Panabacillus mobilis is in everything.
Yes,
Every single
That's
one,
right.
that's what I was going to say.
Except for the field flax, huh?
which is the control. Yeah, exactly.
So that's pretty wild. I'm just looking at
I
Well,
thought-
flax is
what-
an entirely different. It's
Plant
not
is a
plant
control.
cultivated.
Yeah,
Yeah.
So that would make sense that because it's not in the cannabis family or Cannabaceae even. I don't
Right.
think flax is in Cannabaceae. I could be wrong.
No.
But that must be like some sort of, I don't know, important microbe to be within a seed for it to be showing up in all of these cultivars. That's pretty
Well,
Yeah.
yeah.
To be honest, I'm too fastidious, I think, to say that like with confidence this was a big enough sample size or whatever.
I agree.
Did
They say anything about how
I think
they
it's
chose their
cool.
sample, Matthew?
Yes, they did, and I think I also highlighted because I knew that we would ask that question.
That was the other thing I thought about looking at that map. I'm like, okay, so they got like one from over there and then like a whole bunch from somebody else. And it looked pretty ad hoc.
I always love going
Yeah.
into the methods because oftentimes you can start to see how big and well orchestrated the research could be.
Yeah.
Yeah, and how much
Yeah, I think they put the methods at the end. This paper was platformed with.
feel like I feel like I read something related to that, but I'm not sure if I highlighted it because I wanted to like
That's funny.
highlight things that people might find interesting over informative. That's because
In any event, yeah,
Social
I
media
don't
too
need
much.
to knock you off. Maybe we'll come to that if you.
oh no. I'm curious now that you say that because I know I did. Anyways, yeah, this part here, they say phylogenetic analysis of the 136 seed-borne cannabis bacterial endophytes demonstrated that a large number of the cultural bacteria, and they make a good point here, cultural bacteria taxa, right? There might be a whole bunch of stuff that's not getting looked at. So that's a bias that we should consider, right? They're culturing these in a particular medium.
So some stuff is
like-
non-culturable for the layman out there. There's things that
That's
might be
right.
in
Most
nature that
things
will never show up in the
they
lab.
say.
Okay, so that is important to
Some
note.
say. hearsay.
Yeah, they're trying to grow something basically, and then they're counting the stuff that grows. And if it just doesn't grow, then they don't count it. It doesn't mean it wasn't there. They might have killed it instead of growing it.
Well, and like
Exactly.
in a tissue culture type germination, like they're doing in these little petri type dishes,
Yeah, in a
it's
petri
different
dish,
than
exactly.
like a pot of peat or some soil or whatever it's planted into. I think plays some role
It
into.
said they did 5 seeds of each, and then they went to great lengths to clean off the stuff to get to the end of it.
Yeah, they did. They kind of did both. I think the way that I was reading it, and this might not have been what happened, but like I think they did it with the seed wash first, and then they tried without. But not that matters, I suppose.
It
Yeah, so
matters
they mentioned
mainly so
that.
that they know what they're measuring, I think.
Oh yeah, no. I get that. Yeah,
Yeah,
just that. I don't know. I think it's like again, kind of like what you were saying with the picture. Like they wrote it story like with a narrative kind
right.
of voice, a little bit. You can kind of uh feel it. But they mentioned that the genera, so Bacillus was in 28.26%, Painobacillus was in 67.39%, made up the majority of isolates. They say Bacillus endophytes were isolated from 80% of cannabis genotypes, while endophytes belonging to the genus Painobacillus were isolated from all 15 accessions. And that in the absence of environmental microorganisms, the cannabis genotypes in the study appeared to inherit a group of closely related endophytes from parental seeds. So they go the extra, line at the dot and say that, as you can kind of see here, yeah, you see a lot of bacillus, right?
Mhm.
I think Painobacillus is also pretty close together in that greater clade. So cool. They did mention here, this was an interesting point, one of the varieties, X59 or X59, they had like 2 different samples. So the first generation, which they delineate here as X59 Gen 1, was produced by field-grown plants in, what's that pronounced, Vegreville, Alberta. Some of these seeds, well, somebody know? Okay, anyways, some of the seeds were subsequently sown in soilless commercial potting mix and grown to maturity indoors in Kelowna, British Columbia. And the seeds produced by these plants were labeled X59 Gen 2. And what they found was that analysis of the endophyte 16S amplicons showed that the strains of Bacillus megaterium and Painobacillus mobilis, like we said earlier, isolated from the seedling endospheres across these generations, were identical despite growing in vastly different environments. So I guess that's because, I guess they're really sure that it's not from a seed coat thing. I mean, I don't, now that I've read it out loud, I'm actually less impressed with the statement. It was better in my head.
That happens to me a lot. I think it's like, oh, it's all impressive. And I read it.
Well,
Like, wait a
no,
second.
they're saying it came from the seed. It was in the seed, not the soil, right?
Well, and it wasn't what
But I
was
mean,
coated
but they
onto
kind
it.
of already said that. I mean, yeah, exactly. But to me, I read this and then it was like, oh, like all the other things that were endophytes. So it's actually, you know what I mean? But you're right, absolutely. Like
Right.
they definitely showed it. I guess they had the data. I guess it added another paragraph, right? Published
I
with
guess it's a
paragraph.
good sign that if you
Well,
get something from somebody who's got like a healthy, whatever plant that produces a good endophytes inside the seeds, then you can be more confident that you don't have to worry as much about washing and going through the stuff. I'll still probably do a slight H2O2 rinse and microbe or whatever, but I don't know. Old habits die hard.
Are you not worried that you're destroying the good stuff that's already there?
That's a good point. I mean, I've done
Or
side by
inhibiting it,
sides,
maybe.
and the H2O2 typically does better in my side by sides than when I don't use it. So I've just kind of stuck with it because it's cheap, easy, and it's really low concentration. I wonder how much it's even
yeah.
really
I
actually
saw,
doing.
I saw a presentation from Dr. James White, the rhizophagy guy, and he
Oh
he
yeah.
was saying it was very difficult to wash off all the microbes that are on a seed because it's just so plenty, and they're in it too. So
All those
they were
nooks
trying
and
to do
crannies.
experiments. Yeah, they were trying to do experiments with seeds that had bacteria and seeds that didn't. and it took a lot to get the bacteria off the seeds that didn't have, they wanted clean. Like, I don't even think
well, if they're
hydrogen
endophytes,
peroxide's
they're
gonna-
not even on it. I mean, they'd be
Right.
But I think he means the epiphytes.
Yeah, right.
Yeah, all of it, any of them, right? To get them down, yeah.
Dr. Coco, I'm forgetting my what seed hull superstructure morphology. I don't know how to say it, but
Oh boy. Okay.
I know it's different for different kinds of plants, right? But
There's a lot of similarity actually, but
there is though, right? So
Yeah.
okay, so then
the plant morphology, let me just say plant morphology, although it's really flower morphology is the most like confusing area of entire sort of animal plant morphology in the world, I think. Seeds are another one, but there's a lot of overlap between different species. So
that's true. So do you know, like there are definitely pores in seeds, right?
in the testa and the seed coat?
Yeah, in the hull.
Not really. No, I think it's pretty
Not
much
really?
it's pretty much a coat that it softens under water, and some testa are have to be physically crushed or have to go through like acids or other things or be burned or whatever. Some
Right, yeah.
are really difficult to break the latent dormancy or the dormant factors of the seeds and then get wet. But cannabis seeds just have to get wet, and the water imbibes the testa or the seed coat and starts activating the metabolic processes on the inside. But I haven't come across anything. I mean, I might be wrong in terms of
I think you're right though. I think you're right.
But
Yeah,
I think it's
'cause-
just that soaking through of the testa, and then it eventually ruptures because it softens.
I can say from
Yeah.
looking at macro photos of seeds up close that I don't visually see any pores. So it sounds like what Doc is saying
Yeah.
would likely be correct, and that the seed is like the shell is a solid seal, but something about water being part of how its natural process, it makes sense to me that would be one of the things. Like some plants
might
that
be-
have to get burnt or other things
Right.
where they're at in nature, it makes sense that would be the thing to activate it, as
And-
well as like a certain temperature. And
There's different kinds of dormancy in different seeds too. Some of them are like physical dormancies, like they won't be able to open unless something happens to them. Others are more of a biological or a chemical dormancy, where the seed won't sort of awaken from its slumber until it heats up to a certain temperature, for example. That's usually not something related to a physical escape from the shell. So there's kind of the escape from the shell happens because of external factors like the seed getting wet, and internal factors like the sprout starting to grow. The sprout starts to grow because it was awakened from dormancy, often by the wetness, but it can also be by these other factors like heat, certain treatments.
I feel like that's
Yeah,
why scuffing and
I
cracking.
think that I was maybe confusing. Like maybe there are some places where there's weaker points where when the hull softens, like you were saying, but I could be just totally thinking of something very specific. I don't remember, but I really appreciate that point. I brought it up just to say that there's probably not movement at all, which I suspected there wasn't, but I wanted to be sure by asking that poor question. So there is really quite a bit. I think they also mentioned in the paper here that like usually, or maybe it was a different one I was looking at. But yeah, there's not a whole lot of organisms, although they're listing a bunch of examples
about
here.
Movement, Matthew. If you're talking about this is an endophyte inside the seed, like it's inside the seed.
It's inside,
didn't, like,
and it usually
invade
gets
the
there.
seed or go in and out or something. But yeah, it was in there from Genesis.
Well, somebody was
Exactly,
asking,
and it's very controlled. Not a lot of organisms. I mean, certainly there are many now, but it's actually a very small selection of microbes that can really make that movement, as you were saying.
JP from NB asks, Xanthanol, is it possible that hydrogen peroxide can penetrate into the seeds and affect the endophytes inside?
No, I don't think so. And that's, I guess maybe if you like let it soften first, I don't know, hypothetically, but I don't think so. I think that also I think peroxide would be pretty reactive. Maybe
Well,
something
and
that
in my
was
case, I'm not giving it near enough that amount of time, and I'm not scuffing pre-hydrogen peroxide, so you'd have to do something like that. And even then, I think it would probably not be effective. From
I
what
don't
Tao
think
was saying, the
so.
people that tried to do it in studies and measured it, it seems like they had a pretty damn hard time to lower the amount of endophytes or epiphytes, whatever it was. It's just difficult to really sterilize the seed at all in the first place. So, but I do think that scuffing sort of makes like an artificial pore, so to speak. So if your seeds aren't taking on the water well and starting to germinate, that or a crack to allow it to get a little bit of water and oxygen inside can be beneficial. So it's interesting how we can manipulate the biology to sort of work with us if it's not quite going.
Yeah, definitely. This figure that we've been at for a little while, it kind of nicely shows the different hemp cultivars and marijuana cultivars. On the left, it shows the bacterial species they found as endophytes. And in the grid, we have it color coded. It's either red, blue, or yellow. Blue, I guess, is showing that they had the phosphate solubilization capability. Red shows that they had this indole acetic acid secretion, so that's a phytohormone. I think it's an auxin, right? And yellow showed pectinase activity, which is the ability to break down pectin, which I often associate with pathogenesis because a lot of microbes that are good at breaking down plant cells have this capability in some way, shape, or form, but the good guys have it too, ironically, to have for the same ability so they can integrate or do some other stuff. So you can see here that that's the spread. A lot of phosphate solubilization with the mobilis, of course.
Yeah, Matthew, like, let's take that mobilis as an example. You see how it has yellow and blue on the first sample, and then on the fourth sample, it has the red and the blue. So in different cultivars, it's having different action. Is that what that's pretty much saying?
Oh, I think that it's saying that might be the case. Actually, this here summary of in vitro phenotyping assays. I actually don't remember the answer to this. The cannabis endophytes were assayed for the potential to solubilize mineral phosphates, secrete IAA, hydrolyze pectin. Blue squares indicate Okay, I don't know. I actually don't know the answer to that.
Yeah,
it must be, right? It must be.
Yeah, you see what
that's
that's
my inference. Yeah.
So at least on those particular plants, that particular bacteria, or whatever it is actually doing phosphate solubilization and showing pectinase activity. And then the fourth one over, that same bacteria thing is doing IAA secretion and phosphate stability.
Yeah, I feel like that must be what they're saying because they're not attaching that to like a different microbe. So maybe it's a different strain, or maybe it's a different context. I'm not sure.
Okay,
it's a good question though. But my inference would be that maybe it has the capability technically all the time, or maybe certain strains exist. I don't remember reading that in the paper, but
right.
Yeah, actually, that's a good question. If anyone else has a crack at that, or maybe somebody in chat. But
I just think it shows some of the interesting elements of nature and how, given a certain context and availability, it'll act in different ways, and it's able to do more than one thing.
Oh, absolutely, yeah. I mean, it would be totally congruent with other research that we've often talked about regarding that. But yeah, so this is kind of cool. Anyone else have any comments before I move on?
I'll just comment that Tao had a good eye, and thank you for calling that.
Yeah, he does.
I just had a general thing that I'm seeing is I'm not seeing any trichoderma in any of these lists, and you'd think that would be all over the place.
Oh yeah, well, they're just talking about bacteria in this particular figure, but that's a good point.
Maybe it's one of the ones they banned in Canada. Maybe that's why they can't send what's it called, recharge or whatever up there. But Canada's really particular about the types of microbes they have, right? They're very heavily regulated. So maybe certain industries can't use trichoderma, and maybe because they're sourcing all the seed from Canada, that could just be one of the things. Because it's so commonplace down here. But I know that they're a lot more strict up there. We really need to Can Grow or somebody else back to one translate these like we called it Guelph or Guelph. It's actually Guelph, but I was calling it Guelph for a while, the university, and all these different Canadian cities. It's always good to have somebody from that great country up north of us.
Canada.
Yeah. Anyway, sorry.
Oh no, I wasn't. No, I was agreeing with you facetiously. Let's see here. These paragraphs, we already kind of talked about this stuff. They did make this point, this interesting little point here that there was an isolate that was obtained from the God's Bud accession that was identified as PM17 penicillium. This is a supplementary table, which I did not prepare. So that was interesting because following a seedling surface sterilization, only a single accession of cannabis or flax, the control, produced culturable fungus on PDA with tetracycline. And that was this penicillium. So that's neat. This paragraph.
Hey, Matthew, are there anything for the growers to kind of take away from this article? Anything that we would be able to kind of change our practices or things to incorporate into our grows?
I think so. I think a big one is something we already talked about, which is that you're going to that perhaps. I mean, I think it's very likely that plants that you're growing have some of these organisms in them. Some spread, perhaps. Perhaps it's similar to this. Perhaps it's much more varied. Again, we're only looking at culturable ones. So I think it helps to just have that context in your head that, because I think some people come at it from a very totally valid sterile approach, and then some people come about it from a very non-sterile approach, and that's fine.
I think that's helpful. And a lot of growers that I work with definitely think that they are not dealing with microorganisms at all because they're running sort of a sterile res and they're feeding, chelated nutrients. And I mean, I often get asked the question of like, is there any role for microbes in this style of growing? I'm like, there's microbes there. Like,
yeah, right.
I mean, yeah, there's definitely microbes there. So that's a cool sort of lesson to take away. Inside and outside, regardless of how you're growing. So I think it's still probably an open question as to what exactly we should do in all these different situations in order to sort of care for those microbes.
I agree with that.
Or introduce them or not, or other of those kinds of actions. But I think that's cool. So all growers should learn that lesson for sure. That there's.
Do you know when?
Being focused too about what microbes you get, and not assuming that you even inoculated something that it took, and that kind of thing too. Because like we see here, there's a battlefield already waging inside the germplasm of your cannabis seed. So like. There can be funny things that happen, and people wonder like, oh, why did this seed not grow? Hey, maybe it could be this and you don't even know. But somebody was going to say something.
I have a fun fact. I just to answer Spartan's question, why is there no trichoderma? Trichoderma was not approved until November 16, 2022 in Canada for use in cannabis or hemp, and this paper is from August, accepted August 11, 2022. So if there were trichoderma, it would have been in against regulation and use. So it's interesting. That's why.
What do they do when outdoor growers have trichoderma from naturally occurring trichoderma all over the whole fucking country of Canada? What happens?
I don't know, but it's not mentioned in the paper at all because I searched it for trichoderma.
I think it might not be. Well, that's not true, because I know trichoderma can be an endophytes in a lot of cases, and there's different trichoderma. That's, the caveat, right? Like, not caveat, but it goes without saying that trichoderma is a genus, and there's like hundreds of different species. But that's interesting, isn't it? Could have been the medium. I don't know.
What brings it up in my mind mostly is because I know that in combo with the Bacillus subtilis, which has been on there, and it showed that they found it in a lot of associations with the seeds. That if you combine that Bacillus subtilis with trichoderma in the soil, it will help. It's not 100%, but I think it was in the high 80% resistance to PM, powdery mildew. So for outdoor growers, Bacillus subtilis is pretty common and easy to find. So is trichoderma. So I was surprised to see one but not the other.
So I want to pose a question perhaps to the chat and also to the panel. I think it'll be a good question that we could rally around, which is how valid do you think it would be for somebody to take a look at this graph of different cultivars in red and strains of many of these are pathogenic genera of fungi and conclude, I don't really want to buy God's Bud or Grandi or BC Big Bud or Altair because this research report showed that there's some penicillium or some cladosporium or aspergillus or fusarium in those seeds. So how valid do you think that is to do?
Well, I was going to go back to the Dax question and say, when I make seeds, I want to push on as much beneficial bacteria and fungus, whatever I have in my, I have at the time, because that can only help things, right? But
but not.
You look at these petri dishes, they're culturing it. It's not like, right? It's more than
but it had to be there.
Right.
So it definitely came from it, if that's what you mean.
Right.
Like they're not going to grow like this necessarily.
Right.
Yeah. Like this
they also said I think they grew it
in
cocoa or peat or something like that
That's
in
a good
other
point,
conditions. So
this is a little
I assume that
exaggerated.
was
This
peat.
isn't gonna.
They didn't mention. They just said the soilless media. It's usually gonna be.
That's a good point.
Good point.
And also, and as they said in the paper, there are some of these bacteria that they heavily suppress some of these fungi. So I feel like in that way, there's no guarantee, but still, do you want to have? But what if you could have just the good guys?
Is this just scandalous? Are you saying they shouldn't do this because it's scandalous or slanderous against the God's blood or something that it has aspergillus?
I think he was asking as a grower, how much do we consider, and how many of these other people have we bought from and not even known? Maybe
kind of in that direction. But could somebody if somebody looked at this, I could see somebody looking at this and saying, dang, well, I don't want to really buy Fin 34 if it's going to have aspergillus in it, or I don't want to buy VIR 577. Yeah.
I mean, I suppose, but I don't think that this report is sort of marketed to those people. I wouldn't put this probably up in your dispensary if you're, along with the bud, pictures of the petri dishes.
Yeah. But just because their God Bud seed had that on it doesn't mean the guy who grew it in Hawaii is going to have that.
Absolutely.
So
right. Really no. And we don't know the sample size, who knows?
Mm-hmm. And chain of custody, as we saw at the Oklahoma Cowboy Cup. Something can go in potentially uncontaminated and then have a bad chain of custody and then come out contaminated.
Well, you know how a lot of endophytes become endophytes? They started off as non-endophytes, not all of them, but a lot of them. They start off as something in the bulk soil. They get attracted or otherwise make their way to the rhizosphere and the rhizoplane, and they become an endophyte, and then they get up in their seeds when they're developing. And so that's one way it happens.
Certainly, but I would say
not everyone's
fairly
going to get
rare,
this.
and
not
then they
everyone's
colonize. Those microorganisms that colonize there then start to become vertically transmitted from mother to seed, probably.
Yes.
And incorporated into the seed, all of the seeds 100%. Of that mother. And then, the female seeds that grow out already have it and they pass it on to all of the seeds that they produce. So I think the de novo colonization is actually really rare. That's one of the comments at the beginning that we talked about in terms of the founder effect, that, it's rare to come across these opportunities to colonize a niche that hasn't already been colonized.
Sure. But I am of course in that example presupposing that they established, because otherwise, everything after that wouldn't have happened.
I think
so I am presupposing that. You're right. But you're right. It's not a vacuum. And so that's probably one of the selection pressures for having those antifungal, bacteria, right? Because I think it keeps them from getting in easily.
If something like this was occurring, in my experience, at least, and I haven't, tested any of my stuff for these types of infections later in the plant, but I'm presuming that it's going to kill it from a seedling. Like the few times that I've seen my seedling have like a white fungal growth or something, and then it got dampened off and died, where the other 8 were fine in the germination, but those 2 maybe had some sort of endophyte because I treated them all exactly the same. They were all in the exact same environment, but a few of them displayed issues and were immediately killed off, not by me, by what I feel like was their sort of a natural doing. But it was like maybe 5 out of 500 seeds that have grown have had this happen. So we're talking like 1%. And it's definitely interesting to consider.
Were any of these plants problematic because of these endophytes? I thought this was just sort of a symbiotic relationship, not necessarily, beneficial.
Well, do you mean
plant,
symbiotic or okay, just symbiotic?
Yeah,
like mutualistic or parasitic? Well, it's a good question, and that's
neutral.
The reason why I posed this, because another point that I wanted to bring up at some point, but I wanted to have a conversation first, which is that as I've said before, there's fusarium species with strains that are beneficial or have been shown to be beneficial or not really parasitic because, because the context wasn't there perhaps, or because they just happen to be that way genetically. And so we shouldn't assume, we shouldn't be prejudiced.
I actually think we should assume that it wasn't. I think we should assume that none of these were, unless the researchers have said these plants couldn't grow because they were infected with aspergillus or something.
There were a couple. It seemed like there were a couple, but yeah, it's absolutely
I think that
correct.
The inference here is these are just things that are along for the ride, and we found them, and they're there. But plants are still growing. I mean, they may play a role or not. In this, but it's not that this is some sort of contamination and therefore these seeds were not viable.
Perhaps it triggers a SARS reaction in the plant and actually makes it more stronger against other pathogens that are occur. Who knows?
Maybe there's all sorts of microbes that are vertically transmitted that are beneficial that you were in symbiotic mutualistic relationships with.
Right.
That and that may be this, or this may just be parasitism that's fairly innocuous.
Yes.
I didn't do my homework, and I apologize.
No, but that's exactly
interesting
the
to
point I wanted
speculate.
to bring up. Yeah.
I think that they weren't directly answering those questions, but for us to sort of think deeper into it, I love to ask those types of questions and ponder, because there's definitely going to be more answers coming out in the future. And this is just I consider cannabis still on the early days of research, and sort of they're finding this foundational stuff so that they have a baseline. So they'll be able to say in 10 years, this is actually, if somebody finds aspergillus on a seedling, they're not going to freak out. This is been something that's been known for a decade or whatever. But these first times you see it or hear it, it might seem shocking or whatever, but it seems like most of the time it's not killing the plant, but on rare occasions certain things were able to.
Yeah, and I want to self-moderate here, where at the top of the hour we can go into the next paper. I can blast through.
I think so.
I agree, but I want to blast through this part because since Coco mentioned it, just they did mention that there was different seed-borne fungi belonging to genera of cannabis pathogens alternaria, and they have a strain here, PM35, Aspergillus PM40, Penicillium P52, PM52, and Fusarium PM60 here. And I wanted to say that they found that there were bacteria and there were fungi that were able to resist, and you should read the paper if you're interested to learn more. But basically, some of them were able to resist these bacteria more than others. So that seems, I think that they also kind of imply that those fungi were more antagonistic than perhaps we were giving, but that's the important thing, to be knowledgeable. Really, that was the whole thing. Okay, dang. Well, cool. All right, well, let's go to the next one.
I like that the other one is also microbial related.
Yeah, right. I like that too. So this is the one that I suggested. This is called Microbial Consortium Inoculant and Rock Mineral Fertilizer Differentially Improved Yield and Nutrient Uptake of Industrial Hemp Varieties. This was published in Industrial Crops and Products this year. So cool. And yeah, again, I'm going to be not garrulous, hopefully. So basically the idea here.
Maybe share the link for anybody who wants it in the chat. I don't know if maybe Tao or
Oh, I
somebody.
didn't share a link, but I did share the titles earlier in the chat. But yeah, somebody should probably link it up there in the chat.
I'll go and pin if I can find that message.
So basically, they're comparing two different cultivars. One of them is called Morpeth for some reason. The other one's called Han FNQ. And they have a control too. And so what they're looking at is they're looking at an experiment between the effects of a consortium of a microbial inoculum, which is MI, rock mineral fertilizer, which is MF, and a combination of the microbial inoculum and a mineral fertilizer, which is represented as MI plus MF in diagrams that we're going to be looking at and that kind of stuff. They're also looking at, and they're all looking at all kinds of different biometrics like dry weight and root size, width, and root length, and other effects like this. And they found a bunch of
fertilizers, Matthew. Or
that's a good question.
Fertilizers.
That's a good question. I'm actually forgetting off the top of my head, but I think they have a control.
All right.
But no, you're right. I don't remember. I think it is just rock mineral fertilizer. We can dig into the methods though.
Okay. Sorry, I'll let you keep setting it up.
Oh no, it's a good question though. I'm forgetting. And I read through it. They also look at so they look at here root dry weight, shoot length, leaf chlorophyll. They look at different elements like zinc, for example, phosphorus, potassium, stuff like that. And they also look at different metrics at different days after sowing, which I thought was interesting. So we have like 30 days after sowing and 70 days after sowing. And they make some interesting commentary about whether certain things kind of mattered or not based on how much time was allowed for, which I thought was an interesting thing to bring up. So,
okay.
Yeah. So they go here. Let's see here. Do we really want to go into this section here? They do talk about trichoderma, the consortium. They talk about, of course, how some of these microbes they have certain effects that we're already familiar with, as like, for example, plant growth, promoting rhizobacteria. Talk about here. But let me go past that. Oh, this is interesting. I highlighted this part because I thought it would be interesting for us to talk about. They say here that in the seed germination early seedling growth trials from a different paper by the same author, we assessed 14 local and imported industrial hemp varieties in Western Australia. So this is where it's being done. We identified two contrasting varieties, dioecious Han FNQ and locally adapted monoecious Morpeth, for seed germination percentage, early seedling growth, shoot length, shoot growth rate, and seedling vigor index, whatever that is, and root growth indices like average root length and average coarse root length. So what do you think about that? They have a dioecious and a monoecious, and they identified one. Yes, and we identified one of them as locally adapted, which is the Morpeth.
Yeah, I mean, that would just be, we stay so clear of these varieties in cannabis.
I know, right?
Because we don't want seeds. But like, there are some varieties that are just prone to be hermaphrodites, what we would call hermaphrodites, which are monoecious plants. So every hermaphrodite that we grow is monoecious because it has both male and female flowers on it. So they're just talking about a local cultivar that like always herms. And for hemp, I'm not surprised about that at all.
So we have
you could select for that pretty easily. It's harder to select for dioeciousness. But the plant's considered dioecious if it tends towards dioeciousness, even if it can sort of physically be monoecious, like, under stress or under other situations. That's still a dioecious plant. And what they're really saying here is this thing just always produces both flowers.
So there's a lot of diagrams, interesting ones here. I think the first one is like the most complete, or it's the last one. They say here that in the results section that Hemp variety had a significant main effect, I think they mean here, on root dry weight, shoot length, and leaf chlorophyll content. A SPAD reading, as I said earlier, at 30 days after sowing with Han FNQ significantly higher than Morpeth, which they say in figure 1. This figure. They also say that Morpeth treated with microbial inoculant had 3 to 4-fold higher shoot and root dry weight than Morpeth and Han FNQ in all other treatments. We can again go to the diagrams after going after that. So they identified that cultivar had a pretty massive role here, despite being treated, experimentally.
The two cultivars just performed very differently.
Dirty the Grower says the one is a
exactly.
Chinese cultivar of hemp, and they can confirm that.
I suspected that with a name like Han FNQ.
Yeah.
If you can get me the characters, that would be interesting to look at for whatever FN and Q are supposed to be in pinyin.
So I'm curious about what we set out to sort of ask about this article was, how does adding these microbial consortia along with mineral rock fertilizer affect the growth? Like within a cultivar, within a variety would be more interesting. I also think it's going to be a bit of a reach, but I assume that there's a positive correlation. Is that shown some, Matthew?
Yeah, there were some. They did make some points kind of related to that. Since you had mentioned that point, I was curious why you thought that they even did it this way. Kind of like, as you said, I had a similar thought as I was reading this paper.
Yeah, they're trying to simulate conditions that actual farmers growing these crops are going to face. So I'm curious what the source of nitrogen is. But
okay,
in a lot of farming situations, you may have a farmer that's trying to grow a hemp crop and they're adding rock fertilizer. And that may be a common thing to do to grow this hemp in this soil. I saw that they mentioned a sandy soil with low nutrient content. So they're adding rock fertilizer. Is this in Australia? If it's in Australia, then it's probably for phosphorus. And then they're saying, well, will it help if we also add this consortium of microorganisms, of microbes, right? So is that going to help with nutrient uptake? Even though we're also adding the nutrients in this rock form. So that's what I thought the original sort of research question was. Is that right?
I think it is, yeah. So I guess they're just looking at two different plants and seeing how.
So they're looking at two different.
Not really
they're going to test this on two different types of plants. They're going to get the monoecious one, it's a local plant, and they're going to get I guess the Han variety. And on both of those, I assume that they're going to run one group with just rock fertilizer and one group with rock fertilizer plus the microbes. And then maybe other groups would get neither, or maybe one group would just get the microbes. Like that would be part of the experimental design.
Yeah, they mentioned here that so they say significant main effects of variety and treatment occurred for shoot nitrogen concentration. And wow, I can't believe I'm so out of
Does it say what the source
here.
of the nitrogen is?
I'm sure that they do. But I'm not sure what it is actually off the top of my head. I don't think it's ammonia, but it might be.
It's interesting because the YouTube video that I was referring to, they did a study where they did 50-50 and then 100% of one type of nitrogen and 100% of the other type of nitrogen. And in one of the settings, the plant just completely died. The 50-50, the plant lived, and like the 100% one, the plant thrived. So it definitely seems to prefer one of the types of nitrogen over the other one. But having both is effective, and altered ratios of both can be effective as well.
Okay, here we go with some of this.
Here we have some diagrams, which is probably less bleak than all the words. This is figure 4. It says main effects of variety and treatment on A and B are root length, and C and D root diameter, and E interaction effect of variety and treatment on root volume of industrial hemp at 30 days after sowing.
So this is just all about the roots. So it had an impact. What do the colors mean? Oh, it doesn't matter. Okay. A, B. Yeah, I need to spend more time.
Is there a key somewhere by the diagram, or are those just the different conditions?
In the caption, they're explaining what A, B, C, D, and E are.
Yeah, I really don't like. I'm used to a different kind of organization here, where you might have had the C, you might have had these metrics down here, but I guess I kind of see how they did that. No, actually,
so on the high end variety there, you know, root volume, that bottom chart. So the control had about one, it looks like. Just the microorganisms was a decrease in root volume. Just the rock fertilizer also was a decrease in root volume, but using both of them together seemed to lead to fairly significant increase in root volume for that cultivar. But when we look at the other cultivar, you know, the data is a little bit harder to interpret. However, using both still seems to be the largest amount of root volume. I don't know if root volume is a very good way to measure overall crop, though. Does it measure anything other than root volume and maybe another set of tables?
Perhaps in hemp cultivation, that is a metric that is maybe I don't grow.
But you're kind of screwing with the
diameter.
Roots. You're giving it rock fertilizer and then these microbes, and then you're saying like, look, you've got more roots. Well, maybe what you did sort of stimulated them to grow more roots because it was causing the roots to die faster, and that led the plant to like sort of grow more biomass of roots. That's not necessarily going to be a good thing. I'd want sort of other indicators of plant health and vigor beyond the roots, since
besides just association, right?
Yeah, well, we're stimulating the roots in these different things in different ways, and so clearly they may respond to that. But what is that doing to the rest of the plant? Because we're definitely not harvesting the roots from industrial hemp. We're harvesting the seeds or the fiber.
Yeah, I feel as though, I mean, for example, looking at this diagram here, D and also B. Yeah, you know, it's just, I guess, less so with B, more so with D. The spread is pretty unimpressive, but we can keep going to something a little bit more.
I don't know. So I'd want to get into that. It's not totally clear.
It seems to consistently be the best or near the best in all of the conditions, though.
But so is the control.
Yeah.
But the thing is, like Doc is saying, could be an example of science sort of being set up to prove their own point. They think, oh, this and this together is better, and we're going to judge that by roots because when we did this, we noticed it had a bunch of roots. So we're going to use that as our metric and then publish.
Do we care about that metric? Yeah.
But it's like, how does it?
Well, I imagine there's other
correlate to growth.
Did they do anything about above ground like biomass or just root biomass?
Yeah, they did. I think it's the more interesting part of the story. So like, one thing we use hemp for is seed, right? And also when you're growing plants, you also probably want them to be, you know, nutrient maximized or whatever, right? So that the seedling grows very well. So here in 3.4, they mentioned that the variety and treatment has significant main effects on the seed macro, which they define here as phosphorus and potassium, and micronutrient. So that's iron, manganese, right? Oh, how am I messing this up? Yeah, and zinc concentrations. Whereas the variety and treatment interaction affected nitrogen concentration and all macro nitrogen, phosphorus, potassium, and micronutrient iron, magnesium, zinc contents, as table S6. On FNQ has significantly higher phosphorus, iron, manganese, zinc concentrations than Morpeth, whereas Morpeth had a higher potassium concentration than Han FNQ. Cool. In contrast, the control had significantly higher iron, manganese, and zinc concentrations than the other treatments, but similar to zinc concentration in the microbial and mineral fertilizer treatment. So yeah, okay then.
Oh, here's where it breaks down. I mean,
Yeah,
that condition is getting smoked by whatever MI or M1 is.
yeah, and it's Morpeth. So interesting how Morpeth and Han FNQ are disparate in the microbial group. Root length is like triple, practically a little bit less than triple the next highest metric. Wow.
Do we get to
That's
a
in
point though, like Doc was
centimeters?
saying earlier, does it get to a point where it talks about the upper growth in the seed, or have we already gotten there?
Oh, yeah, actually it does. Yeah,
I do
because they
find
have
root length
the
interesting, and it is, I think, an important part of the plant if it's healthy and growing. Root
we
length
want
and
to
root
see the
mass.
upper, we want to see the upper stuff for sure. Yeah, they had a few metrics that they were looking at. This leaf chlorophyll content isn't quite the same thing as like a biomass. They have a shoot length metric here, but I guess that's from the previous paper, actually. That they're like comparing to? Huh. I'm pretty sure they had this information. But no, I guess they don't have a diagram of this. Huh. Well, my bad.
I will say a small subset of people do grow or use the roots, but not primarily. It's more of a byproduct, and they use it for like salves and other things like that. But like Doc and many others have mentioned, the purpose for growing the hemp is either for the seed or for the fiber, the stalk. And it is interesting that that thing that they chose to measure was many people will say the bigger the roots, the bigger the fruits, right? But it would be nice to also have measured the fruits there in this occasion.
Yeah, I wouldn't, yeah, I wouldn't marry the two. I agree with that. Like, I want to see that biomass data for sure.
Yeah, no, if that's what you're setting out to study, but I mean, they might, like I said, I didn't read this. I hate to sort of like drag a paper that I haven't gotten into and really see what the authors are trying to say about it. But
Let me see if I can find that answer though for
it is interesting, and it's interesting that it doesn't appear to be as strong of a relationship as I would have thought.
Here we go. Nitrogen. NH4. Oh wait.
So
they use a mix, is that?
yeah. So
Okay.
that's just a mix of nutrients. Do they give this to all of the plants, or including
It's like a prepackaged fertilizer.
Yeah, it looks like they even got the names here.
Yep. So was that also given to the control? It must have been. I mean, the controls
must have been, right?
Randomized complete block design with 6 replications.
And then the rock fertilizer was in addition to that sort of standard NPK fertilizer, or whatever they were giving to begin with. In
certain conditions. So they had the control.
And this is Australian, right?
This is Australia. So, I mean, if they're trying to replicate the scrub.
Yeah, well, there's just, I mean,
I just feel like fertilizers aren't the way to do that.
Phosphorus or potassium, but like Australian soils are famously short of, it might be potassium. I said phosphorus earlier, but like they have issues with that. If you ever get like a plant that was from Australia, you can't give it too much. I guess it is K, potassium, because they're evolved to like use every little tiny bit that they can get because it's so scarce there. And I think that's probably contributing to some aspect of this research, is like some of the challenges of growing crops in Australia because of the local soils. And, I imagine that the rock fertilizer may be a supplement for that, or I don't know, I'd want to get more into it. But the other thing that I wanted to bring up about the seeds or fiber, which is something we always got to take into consideration when we're looking at hemp studies, is that's not what we're growing with cannabis, or like medical hemp or whatever you want to say. If we're growing flowers, it's a different part of the plant.
But I will say I will make a caveat there that I think that this kind of information is helpful, perhaps from a cultivation perspective where we're maximizing certain seed traits that perhaps, but I was going to say maybe if you wanted to increase certain seed traits for people who are growing from seed. But you're right.
I think that growing
not for the grower
seed,
growing seed, maybe
growing for seed is more similar to growing flower than growing for fiber.
Like a breeder, for example.
Big giant stalks is a lot different than because when you grow for seed, you're still getting a bud that is sexual maturity, and you're producing lots of healthy, full formed buds. But with hemp, for like the fiber, the stalk, they grow it super tall, really, really narrow, like planted really high density, so that they just grow super high, and they pick off all the lower shoots, so that they get these giant stalks. And it's a different sort of goal, like Doc is saying, from the outside.
And even with seeds, Jack, I mean, I think Matthew's right. Some of the stuff, if you want to sort of step up your seed breeding game, and there were ways to like sort of, I don't know, fertilize seed bearing plants better, I could understand that. But that is sort of its own issue anyways, and may put the plant to sort of use things in a different way than it would if it was growing seedless flowers. Nonetheless, I think that this is interesting. My biggest regret here is that I didn't sort of study and didn't do my homework in advance.
You were right about, I Googled what is Australian soil low in, and it says it is known that Australian soils are low in phosphorus and soil pH.
You had it right.
Soil pH levels,
I doubted myself,
could change in response to small changes in the water balance. So it's notoriously difficult just for using a lot of native Australian soil. So Doc, your gut was initially correct. We got to trust our gut sometimes.
Sometimes we do. But I think that that's probably, I mean, at least it's something to consider with studies out of Australia, if that's sort of part of the issue. But I think
you're right that was part of the context.
I think the takeaway from this is that even if you're using rock fertilizer or other inorganic fertilizers, you still might benefit from, microorganism, and you do definitely benefit from microorganisms. Whether or not adding microorganisms, again, how to operationalize this might be difficult, but we still depend on microorganisms in the rhizosphere to help with nutrient uptake regardless.
I
very eloquently said.
Go a little synganic in my cocoa and throw some beneficial microbes in here and there. And I always felt like it would provide some of that stuff that wasn't, although a lot of it is chelated and immediately available, if I didn't have it perfectly dialed in, it would seemingly unlock some stuff and make the plant a little bit happier for whatever the reason was. And not to say that it's easy to get dialed in perfectly, but a lot of people I've seen that implement that tend to be happy with the results. So it's one of the many options out there. And I think that as much as we might try to do a sterile type grow in cocoa, rockwool, whatever, there's going to be microbes involved. And that's just nature. Yeah. Nature kind of finds a way.
You should try to be sterile against the things that are going to cause problems, not necessarily sterile against all forms of life. There's a lot of bacteria that live inside of you that you absolutely depend on. So we don't want to sterilize ourselves either. We don't want to fully sterilize our plants. We do kind of want to run sometimes a sterile reservoir or something like that, though. And I don't think those are necessarily impossible goals to reconcile.
Well, when we were talking earlier about like some of the scarier sounding ones, like aspergillus or whatever else that was in the plants that was found, it's like, I just wonder how many of the plants that I've been exposed to or grown myself had that kind of thing, and I just never knew. And I learned stuff from Matthew about the evolution of people and how like different viruses and things that we've been hit by over thousands of years go on to live with us, and that's part of how we evolved, certain like traits and parts of our bodies and things that we all still live with today and benefit from, despite at the outset sort of seeming like something that would be kind of scary or worrisome, but ultimately it was a benefit. So it's important to look into the sort of reason why it's there and understanding more about what it might be doing or might not be doing and the concentrations and all that stuff. So I'm glad that I learned a little bit more about what is within the plants that we're all growing and loving, and going to be more curious to see what role does it really play, and is it going to be on every plant, whether it's in Canada or in Jamaica or in Morocco. Like there's going to be so much more research coming out. The YouTube video that we were looking at was like Israel study, and they were saying that they had some of the first legal research being done, and it is cool to see kind of where they're coming to. I had to sort of laugh because half an hour in, one of the things they're like, oh yeah, we noticed that the top buds are more potent than the middle and the bottom, and they're like, we tested them and our tops were 12% THC and our mids were like 8 and our bottoms were 6. So they're like, if you take a top bud, it's going to be twice as potent as the bottom. I'm like, okay, that's an important thing to know, but you're growing 12% buds. So like, you're definitely like way behind. And they're like, oh, well, we were doing this because there's no other research at the time. And this was like, less than a decade ago, some of these papers that were coming out, I was like, I remember in the California legal market, there's people growing 30%ers back then. And it's just interesting to see.
But even those kind of conclusions require so much understanding of the experimental setup. Like, how big was this plant? How much light was it under? Like, how deeply under were the unders, and how many other unders were there? And what was the canopy size of the plant? What was the weight harvested off of the plant? I think those two numbers are going to be, I need to know those. If you're going to tell me that your lowers are way inferior to your uppers, I want to know, how big was that plant? How much light was it under? And what was the total weight that you pulled off of that, uppers and lowers? Because I suspect that there was just like too many lowers and sort of all these other factors that could play into that, right?
From the images that I saw in the presentation, and I kind of screenshotted one of them and sent it to the group, and maybe I'll try and screen share this for the Sheep Home Grow faithful out there. All right, we're going to do share screen, Instagram chats, boom. Okay, so this is Nirit Bernstein. I can't pronounce her first name properly. I don't want to butcher it, but this is one of the plants here. And yeah, I don't know. It's looks like it's in a fairly small pot. I would guess maybe 2 feet tall at the most. And there's one apical bud with like 3 or 4 lower buds. And this was a plant pulled out of like a greenhouse full of hundreds of plants, similar sized. And something on the right side, I can't really, even in the YouTube video, it's like you can only zoom in so much. But this plant looks a little bit bigger, but kind of it's held up by bamboo stakes and looks a little bit kind of wildly grown. But the majority of the plants that I saw and the rest of the research looked like this bud here on the left that was just in a small pot. Looks like a short veg, quick flip, just a bunch of plants, and never got them really super huge.
Okay. Do they say anything about the lighting?
I think it's greenhouse, maybe artificially like assisted.
Okay.
But I'm pretty sure that it was meeting a certain lighting thing based off of whatever they might even give them different conditions. But
if it's greenhouse, then the lighting is probably going to be inferior to most indoor grows. I mean, if you calculated the quantum load in terms of like the cumulative DLI across the flowering period, it's usually less. It's just cheap, because it's greenhouse light. But yeah, that's going to be an issue. Like, if you're not providing enough light to your plants, then you're growing a lot of bud off of them, then I definitely think that uppers are going to be better than the lowers.
So you can see here's another example, sort of a bunch of small pots, very short plants, pretty much single buds, just growing up relatively unmaintained. There's no real plant training or anything being done. It doesn't look like they're de-leafing. It looks like they're probably just automated watering. Later on,
are there side branches coming off of that, or if not, they're trimming something?
Maybe they're just removing any of the side branches. But somebody, Wiener DWC, sent me a chat, and the chat has a bunch of timestamps, and like later, like at 49, they were talking about like potentially they were saying nutrients doesn't cause hermaphrodism in their experience, but they were saying that like if their watering system like crapped out, that ended up causing their plants to grow male flowers.
Yeah. Like drought stress, basically, or salinity stress.
Essentially, yeah.
Yeah.
I'm just going to kind of jump through so we can get an idea of some more of their plants. I think they even showed like environmental stress. They're talking about heat and stuff. There's another example of that plant here. It says light, temperature, stress, mineral nutrition, yield quantity versus yield quality is sort of what this is talking about. The title of this video.
They
It's
both
from
say
Cornell.
quantity.
Oh, okay. Yeah, they do both say quantity, but the actual title, I was going to say, getting down here, N. Bernstein, the power of nutrient management for optimizing cannabis yield, quantity, and quality. And she ends up talking more about, she kind of makes the argument that if you want to yield like less, you can get higher quality, but at the same time, it's better. Yeah, she's also saying like if you yield more larger plants, ultimately you're going to get, even though it might be a lower percentage of cannabinoids per plant, if you're just getting so much more, it comes down to that trade-off of.
Is this per plant or like per what? Per square foot?
I think there's, it could be applied to anything, one plant, however many square feet, but ultimately I would always advocate everybody tries to grow their best, healthiest plants. And they're saying in certain situations, even if you don't, you're going to have your percentages start to go up. They measured CBD, THC, CBN, CBG, all this different stuff. This is what I was talking about earlier. The bottom, center, and top, and their top bud A is 12% versus their middle are 6%. And the other condition is even worse. The top bud is 6% and 4% and 2%. So it could be a thing with the strain there that they're growing as well. But I'm going to keep on jumping through. I think it's interesting just to sort of
And what
highlight
are those
maybe,
percentages? I can't see that very clearly, but is that 0.2 and 0.02 rather than 0.04? Or
so on the left
where's the
side,
decimal on the CBD?
oh, at CBD, yeah, that's 0.00, 0.2, 0.4, 0.6, 0.8, and 0.1.
So it's 0.8. It's not 0.08. Okay. I couldn't see that.
0.08 is
No,
correct.
0.0.
Oh, it is. It's
0.08.
0.08. Sorry, I made it bigger. So
0.0,
that's a
yeah.
tiny amount of CBD too.
Right.
Tiny.
Which is
Like
common
the
for like
top
a
of the
THC
scale
variety.
is 0.1.
It just shows me that I would guess that this is just a not so well grown THC variety. I haven't actually figured out what it says. Independent crop production 129, 1581. So this is from 2019, people, as a reference. I mean, it is 4 years ago, but I know a lot more information was out there than maybe they were made aware of.
Wasn't one of the cookies crosses.
It could have been, who knows? Even, good stuff can be grown poorly enough, but they also examine roots.
You're chalking it up. I thought that they were good growers. I was checking it up to.
I think it's a combination of
variety.
genetics as well as cultivation. Because like looking at these plants, like you see this one on the far left, is clearly struggling. Root mass isn't very good. The leaves are kind of crunching at the tips and curling over. The plant is tiny. I think this might be at day 30, 80, 160, 240, 320. Wow, this is it.
That's a 30-day-old plant. Yeah.
Yeah, that's exactly, Doc.
Okay.
I do think that it's interesting if anybody wants to listen through it. I don't want to obviously go through and listen to everything, but they look at different. The one thing I did appreciate about the paper, I thought that was done well, is at this point they look into each individual NPK nutrient and look at how little you can get by with and how much can be applied while measuring certain things. So the different concentrations from like 15, 60, 100, 175, and 240 ppm. I think this was just fairly well done, these parts of the research.
So is that why the plant was so small on the right, is they were sort of starving it of nutrients?
It could have been, but I think, to be honest, I think that was actually one of the stages where they were just trying to grow it in their like field crop. gradients in the medical cannabis. So you see here, this is like, okay, this gives us an image. We've got lighting,
Yeah, it's greenhouse.
but it's not high
supplemental.
intensity lighting. That's just like you're saying.
Yep.
To maybe keep the photo period until they're ready to flower them. And then I think it's probably pretty much just whatever light they're getting in wherever they're doing this research. I believe it is actually in Israel. So I'm not sure what parallel they're on as far as like latitude or whatever. But I don't know. It could also just be there's probably people in Israel that are killing it with their sun grown, but maybe this group just doesn't have it as dialed in. But
well, regardless, with a greenhouse situation, you're going to have light at or above 1,000 micromoles per square meter for a few hours a day on a good sunny bright day. And during some part of that, on a really bright sunny day, it's going to be so high that the plant actually has to start turning away from it because it can't use it all. But that's only for a few hours. And then light in the morning and light in the evening is relatively low ppfd, so the plants aren't going to sort of be able to harvest as many photons over the 24 hours as they could in the grow tent. This is one of the big advantages of indoor growing, is that we can provide like the full optimized level of light the entire photo period. You just can't duplicate that in an outdoor in a greenhouse situation, because you can try to supplement the lighting, but you have to use quite a bit of it during those shoulder seasons, the dawn and the dusk.
I think this is a lot of their foundational research. I think they're one of the first people who got grants over there, and they're trying to, it seems like when they went through online stuff, they couldn't really figure out what to trust, because a lot of the stuff online, like many who have gone out there and tried to figure stuff out on their own on the internet, will find it's a crapshoot. That you ask 10 growers, you get 11 different opinions, is a quote I saw the other day, and I laughed at, which I think is funny and true. But it is tough for a researcher who is in an area that for a long time didn't have any cultivation and strict regulation to go into a time where it's pretty much free to do whatever you want, and how to best take advantage of it. This is looking at nitrogen concentration, which is kind of cool. I think it's 30 part per million, 80, 160, 240, and 320. So the different ppm.
Yeah,
I think that they said 160 is their idea.
yeah,
It looks a little bit, maybe a little light. I think 240 looks, it's interesting there. I'd say 160 is probably fine. I wonder what I'd have to rewatch to go under. Oh, okay. So highest concentration of secondary metabolites was when they kind of starved it, is what this was saying. But you can tell that is just clearly a much lower yielding bud than 320, 240, and 160. Just visually, the plant, the size of the inflorescence. So it's sometimes I am weary of the conclusions that are drawn of early research, because you can measure something and be like, wow, this is a really interesting finding, and then lead people in the wrong direction. Like the drought stress thing, I always try and advocate that you shouldn't do it more than one time. And oftentimes people just do it once on accident throughout their flower. Maybe their watering system doesn't work, or maybe they don't get to their plants in time. So they shouldn't ever intentionally try and drought stress if they haven't really dialed in their system already. But I think that this stuff can really fun to look at. And you can look at some really cool diagrams like the ones that are on the screen and try and draw some interesting conclusions. Here's the effect of K fertigation. And some of the stuff kind of corresponded with some things I think Spartan actually said on a podcast years ago that I heard he talked about. I can't remember if it was K or P or something else, but one of them, even if you gave it not enough, it looked like it still was able to perform fairly well. And it might've been K, but look, 15, obviously struggling. 60, it seems to do fine. 100 and 60, not too much difference. 175 and 240 both look kind of overfed to me. So I would guess they said 100 ppm is where I think they're saying is the ideal. But I'll have Spartan jump in because he's been fairly quiet over there. And I remember that maybe you still remember the study that I'm referring to, but you mentioned that a long time ago that cannabis can kind of go without one and still perform pretty well.
I would say phosphorus. I was saying that people put too much weight into phosphorus usually in the flower cycle than what I think needed. In that if you were going to put any weight on anything, I would actually put weight on K. I'd put weight on potassium in flower over phosphorus in my situation, and in most cannabis fertigations and stuff that I see, and like that's the way I usually go.
Except cocoa.
Yeah, I don't. Yeah, that's true. Yeah, except
Cocoa's
cocoa.
got a lot of K. We often, and that's sort of the issue when we use bloom boosters in cocoa. And I recently released a new version of my nutrient chart where I radically sort of toned down the level of PK boost that I was recommending, because it leads to cocoa growers having like K tox or symptoms of K tox, too much potassium.
Yeah,
So I just wanted to throw that out. I agree with you for the most part. I just wanted to say that about.
And I know I agree with you too. I wasn't really thinking about the, you know me, I'm usually thinking on the organic side of things with Pete, but yeah, for sure. And
You can be
that
organic
can get
in cocoa,
dangerous.
too.
Organic cocoa. I don't see that.
Spartan has been. He does the cocoa loco.
Organic or non-organic. I think there's other aspects to that, but yeah.
I think typically people think of the fertigation in cocoa, but even Spartan himself has done cocoa loco, which is the kind of dry amendment
Organic
version, which
There's
I think he said worked well.
organic ways to fertigate.
Yeah, it's
Yeah.
just a different process of how the nutrients broke down and taken up by the
Yeah.
plant.
Yeah, or whether you depend on them to break down what the sources of them are. But I
Look at this though,
I don't
30
think
to
that's
90
necessarily the
ppm.
point that we tried to raise anyways, but I didn't, cocoa could certainly be run organically.
So Spartan,
Yeah.
was it P that you were saying you can get away with not boosting too
Lower.
much and getting
Yeah,
using lower?
yeah.
They said 30, and they're like, you really don't need to go any higher than 30. I remember them talking about this part. 90, I think, is just like a to make sure or whatever. But any higher than that, and you're really kind of just using too much just to use too much, from what they're expressing at least. And looking at their different side by sides, it's always interesting. The DQ I saw earlier is Dessert Queen, and RM is something else. So there's some semi-modern strains, I believe.
What was the ratio? It was basically, I thought 240, 130 was what they're saying for NPK.
I think so. So we've got
and
30 to
P
90
is
for
the
P.
30. Yeah. What
And
was that?
then-
I
230?
think it was 100 for K.
K was 100.
K is 30 also, I think.
No.
30 ppm for vegetative stage. So let's go forward a little bit. I think they might have gone to 100.
Yeah, I was gonna say
30 to 90. Oh, that's it went too far.
That's P.
Did you say dessert queen or desert queen?
Desert Queen. I read it
Okay.
Dessert Queen, but it is Desert Queen, right? Because dessert has double S.
Yeah,
double-
although
You
both
want
are
two
great
of them.
names.
You only want one desert, but you want two desserts.
Yeah.
Bilber says there's an extra S to make your ass fatter.
Yeah,
That's how I
yeah.
love it.
Find the flowering of P and K. That I want to know what that is, because that's whack. If it's
When
only,
we're looking at it.
it's got to be different. That's vegetative growth.
100 for K,
Yeah. that's
for
what I had.
reproductive.
And there's 100 for the K. But phosphorus, so I wonder what they have that.
Nitrogen was 160.
Oh, just 160 for N.
Yeah, you see, that doesn't even make sense to me.
It
From
seems
all
low.
the commercial available cannabis nutrients, it's like 5, 11.
Also, what
20.
ppm are they fucking using? That is throwing me off now, because I just remembered that there's 3 different ppm scales.
No. Hold on. Can I
It's
talk
still
about this for a second?
ratio.
There's
Yeah.
not 3 different ppm scales. ppm is parts per million. When you measure electrical conductivity and you're trying to guess what the ppm is, there's 3 different ways to guess what the ppm is from measuring electrical conductivity. And there's different assumptions that they make in basically different parts of the world about how much calcium there's going to be in the water, because the little sensors that we use all measure electrical conductivity, and then they report to you a value in parts per million. What they're doing here is they're actually measuring the molecular weights to be 160 parts per million.
Yeah,
and
milligrams per liter.
Yeah, and the effect, interestingly, of 100 ppm of N on EC is going to be different than 100 ppm of calcium on EC. And the 100 ppm of calcium is going to have twice as big of an impact on the electrical conductivity as the N is. There's sort of a relationship between them, but this is the correct way to use ppm when you're actually measuring the molecular weights and referring to it as parts per million for that specific element. I hate all forms of measuring electrical conductivity and then talking about parts per million. And I wish growers would stop doing that. Just if you're measuring electrical conductivity, we should just talk about electrical conductivity. It doesn't make sense to translate that to parts per million because we don't know if it's N or K or calcium or magnesium or what is in that. And when we're talking about parts per million, we want to do it like this. We want to say, you know, you want 30 to 90 parts per million of phosphorus. Aggregate parts per million.
It's an exact number. It's an actual number.
Yeah.
I forgot that. I'm so used to talking to growers that are measuring with an EC meter and then having a thing transfer.
And then
I
about
asked
parts
them,
per million.
What
and that's just the wrong way to use parts per million.
It reminds me a lot of the like ambiguity of like watts and light, right? Oh, it's a 300 watt light. That doesn't mean you don't know exactly what that means, right? It's like people will do it. It's not exactly the same thing, but I share a similar distaste for.
A lot of people assume it's pulling 300 when it might not actually be pulling 300. And
they used to just lie. Yeah, they used to
say
true
too.
That's true too. Would only pull 200 watts.
I got a question. When they're doing this,
before you ask Sal, I just want to give Spartan Grown the opportunity because I know we're about at that time to he's gonna go take care of the dogs and get ready for Michigan Bros Grow Show. So Spartan, any final thoughts and shout outs before you get running?
Yeah, just I just want to thank everybody on the panel and everybody who came out to watch the show and chat. Chat was had their own kind of thing going on too, so it was good. And we had some good input from chat too, I thought today on some of these, especially this last one. Looks like a couple of these guys might have been some of the people that recommend watching that one. We Nerd and somebody else were saying that there's some errors in that we have to watch the whole thing to get the whole gist of it. Some of those slides they had switched. So that's interesting, but thanks for suggesting the video, guys, in chat. And I just want to encourage that if anybody sees something cool that you think we should talk about, let us know. I like that idea. I like the idea of having more eyes and ears out there to bring in more information to talk about. So I thought that was pretty exciting today. But yeah, I'm getting ready to go up to the Michigan Bros Grow Show, and you can catch me there in about a little less than 15 minutes. And we run that show from 9 to 11. Other than that, we'll see you guys here next week, and I hope you all just keep growing.
Peace out, Lord.
See you guys.
Always a pleasure, Spartan. Thank you.
All right, so they're doing the variable amounts of K, P, and N,
right?
But so they'll show K going, from 80 parts per million, whatever, to 160. But what about the other two? Where are those? Like,
while they're running those experiments, the assumption, and they should talk about this in methods, is that you hold everything else that you can hold constant. So like once they determine that the right level of N is 160, then they would test all the K concentrations with N held constant at 160 and with P.
This is what they did.
Right. But
this is exactly what they claim to do.
So
that's what they claim to do.
Yeah. If they're saying N is perfect at 160, what was P and K when they figured out N was perfect at 160? Because
they used to the best agronomic knowledge.
You know what I'm saying, right, Doc? Yeah.
They get it close from hemp.
You got to chase your tail. Yeah, you got to find your tail somewhere and just go from there. And you may need to go back and readjust your assumptions at the beginning when you do it that way, but you start to learn.
They get pretty darn close though, as you saw.
Keep going in a circle, more or less, I would imagine, if I was going to try.
You sometimes have to just refine it, and you realize, oh, N is really too high in all these. Let me retest now that I've realized N is too high. Let me go back and test the different concentrations of K at this new concentration of N.
Right. And then
you're like, okay, now I got K. Let me go back and make sure that the N is correct now.
And the other one works, right?
Yeah. So you just go back and verify. But
I need a scientist. I need a lab and some underlings, about 100 underlings.
Yeah, that's about right.
It's a good point though, because in order to do this really carefully and really accurately, like you do have to be careful, and you do have to run a lot of different experiments, right? With like just to test that this one little thing didn't change anything.
Right.
They threw a crap ton of money at this, and they did a lot of testing. I will say I respect this portion of the research, even if they're not the best growers on the planet.
Yeah,
you can see they put a lot of work into the magnesium concentration, to the nitrogen concentration, and they tried to the best of their abilities to dial it in as well as they can. and at least it gives them a baseline of a place
Right.
to get their
Yeah.
research to continue to move forward, and they'll make subtle tweaks and variations going forward, like nutrient companies, like our home growers out there, like
Yeah,
other
everybody's
scientists who
always
have Bugbee
investing in it.
to others who have labs here in the US and Canada. It's really, I'm fascinated to see it, because I do believe that this is kind of still on the earlier side for them. And I think that this is one of the situations where the home growers and cultivators sometimes will be able to turn out, maybe a better looking crop, or a crop that tests with higher percentage in the desired cannabinoids and terpenes and things like that. But soon enough, trust me, these scientists, they're very specific, and they know the agronomics. They'll start testing the how many inches per day, or millimeters, and they look at all the different number of leaf tips, and they start calculating all this stuff and putting it in Excel sheets, and they can optimize and dial it in and start to improve their systems over time. So I'm definitely a believer that this is going to lead
That's
to
why we
positive
like
things for all of
watching.
us.
Yeah, that's why we like watching these scientific studies and reading them and staying abreast, because they figure out new shit all the time.
Stuff's coming. I want to echo that too. Yeah, like stuff's coming. I can see it, sort of, that there's going to be products coming from some of the exciting science that's come out over the last few years.
Like this one has a 120 ppm range, for example. They're at least honest enough to say, hey, we found between 20 and 140 ppm was what they found to be optimal in their magnesium. And that could be strain to strain. That could be the concentration of, whatever in this particular research was done. But at least they're giving people a range and not just saying, hey, this is the number, follow it. This is obviously the best one. It's like they're showing what they think to be forming in their research. So I got to take my hats off to them.
This is really more, most of us don't mix elemental nutrients though. So that is information that nutrient companies need to be sort of aware of, whether or not the formulations that they're able to keep stable arrive within that optimal range, or how close to the optimal range that they're able to sort of arrive with their different blends. But most of us that are mixing nutrients, or they're providing nutrients, aren't going to be mixing it on the elemental sort of quantity scale like this.
It's much more convenient not to. When I first started, the guy I was underneath who did RDWC, he was buying the raw nutrients like N, P, and K, and he was dialing it in, and we figured out really he was just over fertilizing, and we started dialing it back. Everything, he just had everything too high, a little bit of everything too high. And
what was the final conclusions on this study, Jack?
There's this is a culmination of probably 500 different studies, but they're looking at the reproductive growth of the bottom inflorescence and top inflorescence with those different ranges and concentration. So THC, you're seeing when it's basically under fertilized, it's not able to perform, gets to a certain level, it's getting better, gets to a certain level, it hits kind of peak, and then boom, diminishing returns. So I think they're going to say this is like your 35 ppm or whatever. All of the cannabinoids seem to hit a peak and then drop after a certain point. So,
but I meant like overall in general, did they come up with like a NPK number and micronutrient number that should be a goal?
Well, those are the things that we're kind of coming to, and this is the other thing I was talking about earlier, the NO3 versus NH4.
A lot of shit. Okay.
Yeah, this is a very, very, very I mean, it's an hour and a half YouTube video that is just kind of glazing over a lot of research that they've done over the past several years. And she's discussing what they researched and what they found. So I felt it was interesting enough to sort of touch on. That's why I kind of left it towards the end. I wanted to see how far that second Microbial article, I was considering not even bringing this up at all, because it's hard to do it justice. I was thinking about playing some of the audio so that she could actually speak and we could play it out to the people, but then it gets into sort of fair use and all that stuff, which I'm sure it would still fall under, but I don't want to have this risk getting pulled down because we played some content or whatever of somebody else without their permissions or anything like that. So just looking through it and evaluating some of this public science that they shared was very interesting. I'm going to click over to the chat just for a second because shout out to Weeder DWC. They gave a bunch of timestamps. So herm through nutrition, 4927, metabolic shift and usage at 5009. And anybody who's watching, or if you're on the podcast, you can go through and just pause for a second if you want to look at any of the interesting points that we Nerd DWC pulled up, mostly from 49 minutes in to an hour and a half in, talking about different grow related things that would be found in the study that were actually after the points that I was kind of highlighting earlier, which is interesting enough. But yeah, we went through I kind of glanced through the first 40 minutes so far, but we've only got 5 minutes left, speaking of time, with this show this evening. So as interesting as this one may be, maybe we'll jump back to it in the future when we all familiarize ourselves a little bit more or get some more feedback on that one. But with that said, there's 4 of us this evening, so about roughly a minute apiece. I'll pass it over first to Dr. MJ.
Hey, it was an interesting conversation. I'm again sorry that I didn't do my homework, and I'll try to do better next time, I promise. I just hadn't even checked the Instagram chat at all and saw that there was articles there pending for me to review. So, but anyways, it was an interesting chat, and I like doing these little scientific sessions. Remind everybody about the upcoming Spring Autoflower Challenge over at Cocoa for Cannabis. It's the Size Matters Autoflower Challenge this year, which basically we're just dividing people up into groups based on how big your grow is. But then the side challenge is going to be to grow the biggest auto or to grow the smallest auto. And we're going to have some cool prizes for that. So I think that's going to be fun just to see what some of these growers can do and set with the task of growing the biggest auto. So come and join us for that. It starts on 420, cocoaforcannabis.com/challenge. And I hope you join me for the Ask Dr. Coco show every Monday night for my Patreon subscribers. So grow love, everyone.
Definitely make sure to check all that out. And next up, we've got Matthew Gates.
Yeah, this was a good analysis, although I look forward to some other ones where we can sort of dig in a little bit more into the diagrams and that kind of stuff. I think it does make things a little bit easier, at least visually. So yeah, and good chat. I really appreciated getting those timestamps. Maybe we should put those in the description. But if you're interested to learn more about pests and plant health and that kind of a thing, you can reach out to me for professional inquiries at zenthanol.com. You can reach out to me on social media. Twitter and Instagram is at SyncAngel. And I have my Zenthanol YouTube channel where I'll be posting an ultimate russet mite video. So you guys should check it out. And I also will be hosting a workshop, 2 workshops, one in New York, one in San Diego, in June and in May and June. So to check that out, check out growcast.com/classes for the workshops.
Good stuff. And last and certainly not least of the panelists with us this evening is the American one.
Jack, thanks always for hosting, and shout out to everyone on the panel. It was really a good discussion. Thanks, Matthew, for taking the brunt of the load this week. That was awesome. And
thank you.
Thanks everyone in chat for hanging. I'm the American One on YouTube and the American One _with_Keens on the IG. Most of you know where to find me. If you search for me on IG, if you put in the American One, hopefully a little guy with an American top hat pops up holding the earth. That would be me. And yeah, hopefully we'll see you next week and see you on a couple of chats during the week. And yeah, have a great one, everybody.
Cheers, Tao. And I'll definitely say check out amyaces.com if you're trying to get some of those seeds. And I also want to second Matthew's statement earlier. When he calls something ultimate, he doesn't use that term lightly. This man puts a lot of research into his general projects. So when it's the ultimate, russet mite, I'm looking very much forward to that, because I know there's going to be lots of research, lots of information, and probably more information than some of our minds might be able to handle in a single viewing. But I'll rewatch those, bookmark times, and figure out the best information, because you put so much research into it, and it's always great stuff. So I'm looking forward to that. And I guess last myself, you can find me at Jack Greenstock on Instagram, Jack_Greenstock on Twitter, 50strains.com. This week we'll be hosting the first drop of Velvet Punch F3 seeds. So if you're in the United and you would like to get some Velvet Punch F3 seeds, they're regular, male and female, photo period. But this will be the first time that they're being released to the public anywhere. So that'll be interesting. But I've got some of them I'm going to keep on packing more and more of them. and looking forward to getting those out to the people. Plenty of people have asked, so you'll be able to find them on 50strains.com. And shout out to everybody who's supported this show. Myself getting books, all the people on this panel through Patreons, YouTube subscriptions, comments, likes, feedback in the posts, things like that. Research, Weiner DWC, going through, giving us all those interesting timestamps that I will copy and paste over into the description for people to go look deeper into the research that we covered in the second half of the show. But I'm very thankful for everybody out there who listens and supports us in so many different ways. So thank you all very, very much. We look forward to doing this every week. I really enjoy the science episodes, even if we don't go super deep into them. I think, scratching some of those surfaces, making people interested, they'll dive into them a little bit more. I'm going to go look a little bit more deeply into some of this stuff, and it just continues to further our knowledge and cannabis experience. So very thankful for everybody tonight, the panel, the chat, and all those who listen afterwards on the podcast. Thank you very much. Jack Greenstock signing out. Peace and love, y'all.
Grow with love, everyone.